Inside every single one of your cells, there's this vast library of information, your DNA, and it's being read all the time. But have you ever wondered how the machinery in there finds the right page, starts reading at the very first word, and then just takes off? Well, today, we're diving deep into the very first, very dramatic moments of a gene's life, what I like to call the Great Molecular Escape.
So picture this. You've got this huge, incredibly complex machine built from dozens of different proteins, and it assembles itself perfectly at the starting line of a gene. It's all revved up, the engine's humming, but it's totally locked in place. It literally can't move. So what's the secret? What's the key that unlocks it and kicks off the race? That's the mystery we're going to solve right now. All right, here's our game plan. First, we'll get into what we call the starting gate problem. Then we're going to break down the escape into 3 absolutely incredible steps: the RNA shove, the DNA snap, and the factor swap. And finally, we'll zoom out and see why this tiny molecular ballet is just so critical for, well, all of life. Okay, let's start with part 1, the starting gate problem. We've got this machine that's perfectly built, ready to go, but it's completely stuck. Now, this machine, it has a name. It's called the pre-initiation complex, or PIC for short. The main engine is a molecule called RNA polymerase II, and it's got a whole crew of helpers known as general transcription factors, or GTFs. They all meet up on a special stretch of DNA called the promoter, which is basically the start here sign for a gene. And get this, one of those factors, TFIIH, acts like a can opener, melting the DNA open to create a bubble for the polymerase to read. And here's the paradox, the really weird part. The PIC has everything it needs. The DNA is open, the engine is ready, but all those helper factors that were so essential for building the complex are now basically holding it hostage, locking it onto the promoter. It can't move forward. So how does it break free? Well, that brings us to the very first step of the escape. And it's not some fancy chemical reaction, it's just a simple physical shove. Okay, so the polymerase, poly2, it tries to get going. It stays in one spot, but it starts pulling the DNA that's downstream into itself. It's kind of like someone trying to eat a really long piece of spaghetti without moving their head. This whole process is called DNA scrunching, and it creates this little bulge of DNA and starts building the first few letters of a brand new RNA molecule. Now, once that new little RNA chain grows to be longer than about 6 units, it hits a snag. A part of one of those helper factors, something called the TFIIB reader domain, is sitting right in its way. I mean, they are literally trying to occupy the exact same physical space inside the machine. It's a molecular turf war. So who wins? The growing RNA. That's who. It acts like a wedge and just physically shoves that B reader domain right out of the way, making it flop around totally useless. But, and this is a really crucial detail, this only knocks loose a tiny part of TF2IB. The rest of the factor and the entire upstream complex is still clamped down hard on the DNA and the polymerase. So, the first lock's been picked, but our machine is still tethered to the starting line. Let's move on to step 2. Because this is where things build to the most dramatic moment of the entire escape, the DNA snap. The key to what happens next is a magic number, 14. Poly2 just keeps scrunching more DNA and making that RNA chain longer and longer. But as that RNA gets close to being 14 units long, the tension in that scrunched up DNA bubble, well, it becomes just unbearable. And then snap, just like a rubber band that's been stretched way too far, the upstream end of that scrunched DNA bubble suddenly rewinds, zipping right back into its stable double helix shape. It's this incredibly rapid, powerful release of all that stored up energy. And the result of that snap is immediate, and it's dramatic. The rewinding DNA whips around and physically smashes into the factors that were holding it in place. This is called a steric clash. It's basically 2 things trying to be in the same space at the same time. The force of this collision acts like a catapult, and it launches the entire upstream promoter complex, TFIA, TBP, and the rest of TFIB, completely off the polymerase. Bam! They are all evicted in one single move. The machine is finally free from the starting gate. Its whole status has changed. It's not an initiation complex anymore. It's now an early elongation complex, or EEC. It's mobile, it's moving down the DNA, but the transition isn't quite done yet. It's kind of like a rocket that's cleared the launch tower, but still needs to jettison its booster stages before it's ready for the long journey. And that brings us to the final step in this whole process, the factor swap. Now, this is a much more orderly event, almost like a molecular relay race, where the initiation crew hands the baton off to the long-haul elongation crew. So we just saw that big eviction event happen right when the RNA hit about 14 units. The relay race keeps going as that RNA chain gets even longer. Once it grows past 22 units, a whole new crew shows up, the elongation factors DSIF and NELF. Their arrival is the signal for the last of the initiation factors, TFIIE and TFIIH, to leave. And then, a little bit later, the very last member of that starting crew, TFIF, gets replaced too, completing the handoff. And this is the final swap. As these new elongation factors, DSIF and NELF, bind on, their presence basically pushes off the last members of that starting crew, TFIIE and TFIIH. It's this carefully choreographed exchange that completely reconfigures the polymerase, turning it from a hesitant starter into this processive, efficient machine that's ready for the long journey down the gene. So we've just watched this unbelievably intricate 3-act drama play out at the molecular level. But why? Why does the cell go through all this trouble? Why does this escape even matter so much?
Let's just quickly recap the whole thing. First, the RNA shoves the TFIIB reader out of the way. Second, the DNA bubbles snap shut, kicking out the main upstream factors. And third, a new crew of elongation factors arrives to swap out the last of the initiation team. A shove, a snap, and a swap. Simple, right?
You see, the critical point here is that this complex, multi-step process isn't just for show. It acts as a series of really important checkpoints. It makes sure that transcription doesn't just start randomly. The polymerase has to actually prove that it can make a stable piece of RNA before it's allowed to escape the promoter. This whole process is a fundamental control point that helps regulate exactly which genes get turned on and when. This elegant dance ensures there's precision in our biology every single second of every day. But it leaves us with a really compelling question. What are the consequences when even a single step in this tightly choreographed ballet goes wrong? Well, the answer likely lies at the very heart of numerous diseases, which just goes to show you how incredibly important it is to get the race started exactly right.